go over some very basic things that you need to know for the class and how to do well in the class. Okay, so this is the book and the platform that we're using. This is the Marieb Anatomy and Physiology. And hopefully everybody's familiar with this because you probably had this during the last semester or the last time you took AMP 1, right? I encourage you to get the ebook version with the package that allows you to access my lab and mastering, because all of your homework, assignments, and quizzes are going to be on this online platform. All right, now it's working. Okay, so you have here 4 unit exams. That is going to count to 60% of your grade. Okay. Besides that, you have homeworks. And that is going to be 20% of your grade. And you have your quizzes. Those are also 20% of your grade. Okay. You add this all up. You come up with 100%, right? You have to know how to calculate your grade. And this is really difficult. Look at this. 60 plus 20 is 80. Plus 20 is 100. Okay. Now, the grade that shows up on Canvas is not going to be your overall average grade or calculated grade. Is not. It's going to be wrong. I'm telling you, it's going to be wrong. Okay? Because the way I have to set up Canvas is in a way that I have to exclude the grade of one exam, because I allow you to take an optional 5th exam, which is the optional cumulative exam. Okay? The final exam. If you do well on 4 exams, 4 unit exams, and you are happy with your exam grade, you don't need to take the cumulative final. Okay? If in one of those 4 exams you didn't do so well, then I would advise you to take the cumulative final. Okay? If you do better in the cumulative final, you can drop that one grade that you did not do so well. Simple? Okay, so let's say in the first exam you had 50, second exam 80, third exam 90, fourth exam 75. So you want to get rid of your first exam grade, right? So then you take your cumulative final, you do better than a 50, so you can dump that 50 grade. Okay, so you can substitute the grade for one exam. If you miss one exam without a good acceptable excuse, then you have to take the cumulative final, because that's going to be the substitute for your missed exam. Okay? If you miss one exam due to a valid excuse, then you have to let us know, because then we will arrange for you to take that exam, to make up for that exam. Okay? Us who? Me and Ray. Ray, you want to say hi? All right, so Ray is our TA, very experienced, very useful, very helpful.
Hello, I'm Ray. My office hours are not in the Canvas page yet, but they will be on Fridays from, does this work? Can you guys hear me? Yeah, okay. From 12 to 1 in the Science Learning Center, which is all the way across campus. It's in the Schmidt something family complex next to the football stadium. It's a great resource also to just go there and hang out. There's VR headsets, there's models, studying things to do, there's microscopes. So yeah, good resource besides coming to my office hours. And also if you just need me at another time that you're available and it's not during my office hours, just email me. Yep.
All right, thank you. Okay. So you have to let us know if you're going to miss one exam, okay? Then we arrange with you to make up that exam. All right, so here, your bonus for being in class. You're going to get in-class extra credit using iClicker. So I know a lot of you are already registered with iClicker, right? And some of you got an email today that you should register and get your iClicker in order, right? So being in class and answering iClicker questions, you're going to accumulate points. Okay? A lot of people ask me, do I have mandatory attendance? No. But if you're not in class, you cannot get extra credit, right? And then at the end of the semester, when you're like really close to getting an A in the class and you're just missing 2 or 3 points, and you're like, oh my God, if I had attended class, I would have 10 points and that would push me to an A. And then it's too late, right? So come to class, accumulate your points, and then at the end of the term, you're going to get up to 10% of increase in your final grade. So if your final grade out of this calculation comes to 85, and you are in class and you accumulate all your extra credits, you can bump up your final grade by 10 points. That would give you 95. Yeah, 85 plus 10 is 95, right? So that would push you to an A, right? Solid A. So is it beneficial for you to be in class? Yes. And do you have to be fully awake and totally know what's happening in the class? Preferentially, right? So let's say that you're just in class because you have to get those extra credits, but you don't really know what's going on. And then an iClicker question shows up and you're like, oh my God, that looks like Greek. And you're like, oh, well, I'll attempt to answer anyway. So just by attempting to answer a question, you get a point. Okay? Even if you're half asleep, zombie-like state. Okay? Actually, I had a professor tell me last night that she was pacing around the class and somebody was shopping for shoes online. Like, okay. So I regress. Okay. So being in class, attempting to answer a question, you get a point. And I think I set up also just for showing up in class, you already get a point just for bonus attendance. Okay, so I'm very generous for extra credit points, right? So here, your extra credit points are going to be calculated only at the end of the semester. I do not update the iClickers like all the time in Canvas because it just drives me crazy. All right. So everybody's is fine on how to calculate your grade? Yes? If you don't really know how to do that calculation using algebra, let us know and we'll explain to you exactly how to do it. Okay? All right. So here is the iClicker class for you to join. Just by show of hands, who would be ready to just start answering iClickers like right now? Okay. All right. So basically most of the class. Who has no idea what an iClicker is? Okay, we do have a few people. All right, so for today, I am not going to give you extra credit points, but on Wednesday, I'll give you the points that I didn't give you today. So be ready to get your extra credits on Wednesday, okay? Either by using the physical remote if you have one, or by using your app on your phone that you can download and do the same thing. Okay, so either physical remote or the app. Okay, and I uploaded this lecture for week number 1, so it is available for you to sign up for that iClicker class. Okay. Okay. So organization of the course. When you open your Canvas, you should go here to the modules. Okay. And you're going to see that the modules are divided into weekly subsessions. Okay, so of course we are here on week 1, and first thing you're going to see here is a PDF file for the lecture. Notice that most of those PDF files still have the course as summer 25. Okay. So I'm sorry, I am notorious to update the lectures like just before the class. Usually it's not a lot of difference from the previous semester lecture, but I always find a bit of a change that I can make things a little bit better or not as complicated. So I always like to tweak a little bit my lectures, okay? So when you check on the weekly PowerPoints, you can base your study on the PowerPoints that are here. And then just before the class, you just make sure that you have the latest PowerPoint that I'll cover during the lecture, okay? So besides your PDF file, you're also going to have a page here that has several resources for you to study, meaning that you're going to find here my YouTube videos, okay? You're going to find the videos from the previous lecture, as well as the video for the actual class. And as soon as we're done with the class, I edit, produce the video, and then I put it up usually at the same at the same night. Okay. So besides that, I also upload here study guides for each of the chapters, and I put links here for the Journal of Visual Experimentation, for the JOVE journal. I usually have my classes access the JOVE site for free. Now, I don't know if I'll be able to get it for this term, but if I cannot get it directly to the JOVE site with the reps, well, I'm hoping that our library will sign up for it, so you're going to have it also for free. But if not, I know it's like, if you really want that, that resource is really cheap. I think it's like $18 a semester or a year, something like that. Okay, but I do like those JOVE videos. I'll show you also the integration with AI to help to study and retain better. Okay. All right, so we go weekly. So you're going to find that you have to go to your Mind Lab and Mastering for your assignments, right, for your homeworks and for your quizzes, all right? So you're going to have here, we're starting at chapter 17, right, the blood. So you're going to have here an assignment that is open. So I will open all of the assignments for the entire course, like today. Okay, so most of the assignments are open. The ones that are not open, they are going to be available, like today or today and tomorrow. So everything is going to be open. And then you're going to have different due dates for the assignments. So usually I do not ask for the assignment to be completed until we complete the chapter in class, okay? And you usually have until this Sunday after the class was completed, like you have until like Sunday night, and more specifically like 11:59 p.m. on that Sunday night, right, to complete those assignments. So you're gonna see here homeworks that are usually a lot of questions, I don't know, 50 or so, and then you're going to have quizzes for the same chapter, and those are, you know, a lot less questions. Now, how is it that that you can get your homework done? When you go to do your homework, you usually have a lot of attempts to get it right. Okay. Here I have about three times per question. I usually put like 10 or so, because I really want to encourage you to do your homework, to go back, to do your studying, and to get your points. Okay. If you have it late, you're going to have a penalty for submitting it late. Okay. But you still can submit it and get as many points as you can, right? Now, yeah, I forgot to turn off my email. So hang in there for a second, because my email is going to go crazy. Everybody's emailing me today. Yeah, and people forgot to turn off their phones too. Okay. All right, so here. For the quizzes, I usually give you 2 attempts on getting it right, right? So you took it the first time and you got a low grade, you take it again, you got a better grade, you keep the better grade. Okay. All right, so here is an example of the link for the JOVE platform. When you click on that playlist, you're going to have a lot of these videos. There are short videos that show up. So what do you do with this JOVE platform? You click on the video, you watch the video, and then you have the option to answer questions that are based on the video. So you get extra practice questions, and there are multiple choice questions that can help you to better understand and prepare for the exams. All right. Any questions about any of that? No, everybody's cool? No. Okay. So let's start with the cardiovascular system. Yeah. No, the Joe videos, those are supplemental. Okay. You don't have to do it. But it's just that I find that they are very accurate, they are informative, and they're just a different perspective on looking at the subject we're studying. Something different other than what I'm mentioning in class. And also, notice that I do have an accent, so sometimes I say some things that you might not understand, or and then you go back and watch a similar video, say, oh, this is what she meant. Okay. But if I say something that you do not understand, ask me, all right? So I can try to pronounce it in the correct way. An example of that is, well, let me ask you, anybody with a Portuguese first language in the class? Yeah. 2, 3, yeah. So what is this? A paper, but you also call it a sheet of paper, right? I have a very hard time saying a sheet of paper because it's sheet and sheet is all the same to me. Okay. So when I refer to this, I refer as a piece of paper. Okay. And there are other words that are even worse, but anyway. But yeah, so I always give you a bit of an alternative to understanding the subject based on the native English speaking person. And Jove is really, really great. I really like that. Okay, any other questions about grading, about homework? Yeah? Sorry, can you? The homework is on the platform that is included with the package for the textbook. Okay? I think you have a couple of options of purchasing it, but I think the Canvas is one of them. I think also if you go to the bookstore, you might also allow you to purchase, but I think it is, you can get it from Canvas. But then if you had it before from a previous semester, you can just, keep putting something in before. Right? Everybody cool? Yeah? Yes, exams are in person using the blue Scantrons. Okay? So yes, you have to come to class. You're going to be in person. Okay? And you have to be ready for those. Yeah. Oh yeah, before I forget. For the final exam, for the cumulative final, right? I allow you to prepare one sheet of a cheat sheet. Okay? So if you opt to take your cumulative final, you can bring to class one handwritten cheat sheet. Okay, front and back, whatever you want, right? But I will stick to handwritten this time. I won't mention why, but it's going to be handwritten. Right, so you have to prepare your own. Yeah. A what? Oh, so you took it when last semester? When you go through the Canvas and you go assess Pearson, it is directly linked to this course. Yeah, so once you go, access Pearson, it will take you to the homeworks and assignments for this course. Okay. All right, so everybody cool? Yeah. All right. All right. So let's get here into the blood, right? So this is the first part of the cardiovascular system. Oh, it's the first part because you actually have 3 parts for your cardiovascular system. You have the blood component, then you have your heart, and that's of course what pumps the blood around, and then you have your blood vessels, and that's what gets the blood that was pumped from the heart to the rest of your body and then back into the heart. So these are the 3 components of your cardiovascular system. Okay, you have your blood, your heart, and all the blood vessels. So here you have your arteries and your veins. Okay. All right, so looking here at the blood, when you study the blood, you study hematology. Okay, so this is a study of blood components and also of the forming elements of the blood, okay, forming tissues of the blood, and also all of the things that can go wrong with the blood. This is studied by hematology. Okay. Now, blood is very important for maintenance of health or homeostasis of your body, okay, because it has some very, very important functions. First thing, of course, is a transport function. So blood is very, very important to transport gases, meaning oxygen from your lungs to your tissues, and carbon dioxide from your tissues back to your lungs, okay? So transporting things like gases also is going to transport nutrients, and is going to transport back from the tissues, is going to transport metabolites and metabolic waste products, okay? Besides that, blood also transports very important molecules that are produced in one part of the body but need to reach a different part of the body for them to function. So this would be an example of hormones. Okay, so blood is really the means of transportation for all these components. Now, besides that, blood has a very important regulatory function that is good for maintenance of a certain blood pH, meaning acidity or alkalinity level, and is important also to maintain the body temperature. Okay, so functional for regulation of the blood has to do with pH and temperature. Now, besides those two very obvious functions, blood is also very important for a level of protection in two different ways. Okay, so first, blood has some elements that can protect you against bleeding to death. Okay, so blood has these clotting factors and clotting mechanisms that can stop hemostasis. Hemostasis means stoppage of bleeding. Okay, homeostasis means a state of health and balance of reactions in your body. Okay, I know the words are very similar, but when we get to hemostasis, you see that the functions are different. Okay, so protection against bleeding, and also blood carries formed elements that are very important to protect you against pathogens. Okay, so the blood has the immune system cells that are important to detect and get rid of pathogens. Okay? So important functions of the blood. Okay. All right, so how thick is blood? Well, when you compare it to water, you know that blood is thicker than water. So that means blood has a higher viscosity than water. Okay, it's a bit more gooey than water. The temperature of the blood is around 38 degrees Celsius. Okay, so this is a bit higher than the temperature that you measure with a thermometer, like when you go to take vital signs and then doctor comes with a thermometer and put it under your tongue or under your armpit, or a rectal measurement of temperature. Usually it's going to be a bit lower than 38 degrees. Okay, because the core blood temperature is a bit higher inside the body than it is at the edges or extremities. Okay. So blood not only is a bit more viscous than water, is also more alkaline than water. All right, who here took basic chemistry? Yeah, okay. Who did not take chemistry? Okay. Who has no idea what a pH is? Okay, that's good. So what is a pH? Concentration of hydronium ions, right? So inverse log of concentration of hydronium ions. But anyway, this is a measurement of how acid or how alkaline a solution is. Okay, when you have a pH that is lower than 7, that means your solution is more acidic. When you have a pH that is higher than 7, that means your solution is a bit more basic. Okay, so blood is slightly basic because it's not exactly 7. If it is exactly 7, then it would be neutral. Okay, so blood is slightly basic, but look at this range of pH values. It goes like 7.35 to 7.45. It's not a huge jump, and it's very important for blood to be kept at that pH, because if it deviates from these numbers, then your body is going to get into trouble. Okay, so you have to think, how is it that a change in acidity is going to get me into trouble? Well, now you have to think what is happening in the blood. What are the components of the blood? What are the soluble components of the blood? And what are their functions? Okay, most things are made up of proteins. Proteins have charged amino acids, not charged amino acids, basic amino acids. Mainly the whole water environment around that protein, if you change the hydronium ions or the charges, you're going to distort the shape of the proteins. If you distort a lot the shape of the proteins, then they're not going to work. That means your body is going to stop functioning. Okay, simple terms like that. So your blood has to be kept at a certain very tight range of pH for all the proteins to work the way they're supposed to work. Okay. And that goes as well with maintenance of temperature. Okay, because if you increase the temperature too much, you're going to denature proteins. You're going to stretch them out. And if you denature proteins that are, for example, in your brain, then you're in trouble. Right. So maintenance of blood pH, maintenance of temperature, those are very, very important functions for homeostasis. Okay. All right. So color of the blood. It can go from quite dark red to very bright red, and that will really depend on the level of oxygen that you have in the blood. So oxygenated blood is bright red. Deoxygenated blood is not so bright. Okay, so the blood, when you look at your veins, you see really on the surface, you're going to see the veins, and they are looking bluish. It's not that the vein itself is bluish, but the blood is more to the bluish side. Okay, so that is the deoxygenated blood that is coming from your tissues back to your heart and lungs to be reoxygenated. Okay, we're going to see more about that later. All right. So how much blood you have in your body? Well, it depends if you're a male or a female. If you are male, you're going to have 5 to 6 liters in average of blood. I know this is America. Nobody thinks in liters. You think in gallons. So what is 5 liters, more or less? About a gallon? A bit more than a gallon, I think. Right? This is when you go on your phone, it's like 5 liters is what? What is the number of me? I always forget, like 4.54, something like that. Something like that, right? So you have a gallon of milk, that's about almost 5 liters of blood. And then you go, what? That's not a lot of blood, right? But then you look at females, and you say, look at this, we have even less blood, 4 to 5 liters. Oh, there you go, 1.5 gallon, 1.2 gallons. Yeah, it would help if I give my glasses. Okay, so 1.5 gallons is not a lot of blood. 1.2 gallons is even less blood, right? Why is it that males have more blood than females? You're going to find out soon, okay? So that depends, of course, in body mass and size, but it depends on other things too, like hormones, for example, okay? All right. So first we're going to look at what makes up the blood, okay? So the blood has a lot of liquidy stuff that is clear, so that is the plasma, and then it has the formed elements, and those are cells or fragments of cells. Okay, so we're going to look at them individually here. So the plasma of the blood, that is going to be actually the plasma in general is going to be about what do you say here, 75% of your fluid that is outside of your cells. Okay, so this is called that interstitial fluid. And then you're going to have also find 25% of all of that fluid that is outside of the cell as extracellular fluid. Okay, so we're going to have more specifics on how to distinguish this. Okay, so here, when you look at the fluids that are outside of the cell and they are bathing the cells, but they are not inside of the tissues. Okay, so you call those that one as interstitial fluid. Okay, so it is not in the veins and is bathing the cells to keep them alive and to bring nutrients and gases to those cells. And then we're going to look more at this when you look at more functions of the blood. So the plasma is made up of basically everything but formed elements. Okay. All right, so here I have this other. This slide is a bit out of place, telling you about the functions of the blood. So just because we're following exactly, you know, the organization of the book. I mentioned all of this already. So so this is for your studying purposes. Okay. Okay. All right, so here when you get more into the composition of the blood, okay, so blood is really the only tissue considered a connective tissue that is liquid in your body, okay? So you have four types of connective tissue, and blood is the only one that is liquid. Now, if you remember from the AMP1, what are the characteristics of connective tissues? So we know that when you look at connective tissue, you're going to find a lot of matrix, that is the stuff between cells. And then you find a cell here, a cell there, like floating around in this matrix, right? So when you look at the matrix of blood, you'll find that the matrix is really that plasma, is that clear fluid that is keeping the cells in suspension, okay? So matrix is the plasma, and then when you look at the cells, those are mainly your formed elements, okay? They're most of the formed elements, not all of them, but basically the cells are suspended in this plasma, okay? Now, cells, they will make most of the formed elements. So here is a description of your formed elements. So basically there are three formed elements that you should focus on here, okay? You have here your erythrocytes, those are your red blood cells. You have here your leukocytes, those are your white blood cells, all different types of white blood cells, okay? And then you have your platelets, those are little fragments of cells, okay? So your erythrocytes, or your red blood cells, those are tiny and they don't have a nucleus, right? So red blood cells don't have nuclei. They also don't have mitochondria, okay? So those are a lot of numbers of them, but tiny and no nucleus. And then you look at leukocytes, you look at the white blood cells, you have a lot of different types of white blood cells. But those are the bigger guys, bigger than red blood cells, and they do have nuclei. Some of your white blood cells have weird shaped nuclei, right, nucleus, but they do have one nucleus per cell, okay? So real cells, you have red blood cells, leukocytes, and then you have these platelets. Platelets are little pieces of cells that came from the cell membrane and a piece of cytoplasm from a bigger cell, okay? But they are also considered as formed elements, right? So for your connective tissue here, remember you have the matrix and you have the cells, and here are your cells and your formed elements. All right, so let's see now what is in the plasma, what is in that matrix, okay? So a lot of the volume of the blood is made of the matrix, a bit more than 50%, okay? And what you're going to find there is mainly proteins, so floating proteins. A lot of the matrix is really floating proteins besides the water. Okay, so you have a lot of water that makes up the matrix, like over 90%, and then you're going to have your floating proteins. So what do you call the proteins that are floating around in the plasma? That's an easy question. Plasma proteins, like right there. Okay? Where is it that these plasma proteins are coming from, like most of them? What makes the proteins and dumps them in the blood? The liver is like right there. Okay, so your liver is going to make those proteins and is going to put them in the blood. Then we're going to look at functions of those proteins in more details. Okay, so mainly the liver is going to make those plasma proteins. Now, why do you need proteins in the plasma in the first place? Right. A lot of the function for these proteins is really to keep this thing that we call osmotic pressure. Okay, so let's look here what that means. So let's say here you have a cell, okay, any cell here. So inside of the cell, what you're going to find? Organelles. You have organelles here, organelles there. Do you have proteins inside of the cell? Yes, right? You have a lot of proteins inside of the cell. In the cytoplasm, you have proteins that are being made and they are kept in the cytoplasm for protein function. You have a lot of stuff inside of the cell. If you put this cell in a bucket of water, what is going to happen to the cell? It is going to swell up, right? So if you have water here without any protein, or without anything here, then the water is going to go inside of the cell because of osmosis. The cell is going to swell up and is going to burst. So if the plasma of the blood does not have any protein, then your cells are going to burst. Okay? So to correct for that issue, now you have your proteins in the plasma, and then which organ produces those proteins, a lot of them? The liver, right? Am I going to ask you something about that? Yes, that's your clue, okay? I will ask you, right? What is producing, the organ that is producing those proteins. Now, if you have approximately the same number of proteins in the plasma than you have in comparison to the inside of the cell, now what is going to happen to the movement of water? Not much. The same amount that goes in is the amount that's going to come out. So you can maintain the integrity of the cell membrane and is not going to swell up and burst. Okay, now let's say that you have the opposite scenario. Let's say that you have too much protein in the plasma. What is going to happen to your cell? It's going to shrink because now the movement of water from osmosis is going to be from inside to the outside of the cell, right? So the level of protein in the plasma has to be kept relatively constant, right? You have certain proteins that have an important job of keeping the osmotic pressure of your cells, okay? All right, so here more on blood composition, right? So a lot of the blood is made up of the plasma. Then you have most of the plasma that is made up of water, right? So look at this, 90% of your plasma is made up of water. Then you have a certain percentage, like less than 10%, that is made up of proteins. And then the rest of it is just made up of some solutes and ions, right? So here, looking at the proteins, look at this. The composition of proteins from your plasma, okay? So most, or over 50% of the proteins are those albumins. Right? Over 50% are albumins. And then you have also a large number that are made up of globulins, so less than 40%. And then you have these other proteins that are the fibrinogens. Okay, so let's see what is the function of those proteins. The albumins, they are mainly working to maintain osmotic pressure. The globulins are the proteins involved in your immune system function. That means you're going to have antibodies and immune defense proteins that are classified as globulins. And then you're going to have this fibrinogen that is important for blood clotting. So this protein can get together and stop bleeding. Okay? All right, and then the 1.5% of everything else is basically very small electrolytes and some amino acids that are single amino acids or gases or waste products. Okay, but this everything else here is a small percentage of your plasma. All right. So this is straight from your book, okay, telling you the composition of your plasma, okay, and here you notice that you have your albumin, okay, and this is important for osmotic pressure. You have your globulins, and remember globulins are important for immune system function, okay, but you also have other types of globulins that are they're important for transport, okay, but we're not going to get into too much detail of that. Fibrinogen is the one important for blood clot formation. All right. So let's see here. So here we're getting to the formed elements of the blood, okay. So remember, formed elements, you have red blood cells, white blood cells, and platelets, okay. So here is your showing a red blood cell. Here's an example of a white blood cell, and those are your little platelets. Actually, platelets, they're usually more round than that. Those are more specifically activated platelets. All right. So when you're talking about formation of those elements, you're really talking about hematopoiesis, okay, or hemopoiesis. So this is where you're going to get your formed cells, right? So where is this going to happen? Basically, it's going to happen inside your bone marrow, and more specifically, it's going to be in the red bone marrow, okay? Where is it you find red bone marrow? Well, it depends. If you're a child, you're going to find it in your long bones. If you are an adult, it's going to be more in your flat bones, okay? But it's going to be in inside of the bones, okay? Now, inside of your red bone marrow, you're going to have this stem cell that can become anything. So that stem cell you call pluripotent stem cell. So when you look at a cell that did not decide what to do, it can become any one of your blood elements, okay? Now, basically, when a pluripotent stem cell decides that it's going to become either a white blood cell or a red blood cell or a subtype of a white blood cell, it is going to take one of two paths. It's going to either go through the myeloid path or it's going to go through the lymphoid path. Okay, and that's going to be in our next slide here. There you go. All right, so I know it looks a bit overwhelming, right? But there are some things that are really important to take from this image here, right? So here we start with your pluripotent stem cell. It can become anything. How is it going to decide whether to go from one path or whether to go to another path? Well, it's going to get signals from the environment, right? So it is going to get certain types of stimulating signals that are going to bind to receptors on this pluripotent cell and is going to activate certain genes to tell the cell to differentiate either one way or another way, okay? We're not going to go into these details here for now, okay? So let's say this pluripotent cell decided to become a cell that is important for lymphoid lineage, meaning that decided to take this path over here. Okay, that is a simple path. Okay, so the cell is going to become a lymphoid cell. So now the cell starts to differentiate, and then it can continue to differentiate, and it can become either a T cell or a B cell or a natural killer cell. So these three cells over here, those are important for immune system function. Okay, so these are white blood cells, right? And they are going, each one of them is going to have a specific type of function for immune system, okay? So let's see here. If the cell becomes a B cell, then this B cell later on can help your immune system to produce antibodies. Okay? If the cell becomes a T cell, then the T cell is going to also help for immune system function, for helping B cells to produce antibodies. Okay? Or it could become a natural killer cell, right? So basically, out of this lineage, you can have T or B or natural killer. So I would suggest that you keep in mind that this lineage only produces 3 cell types, right? Why? Well, because if those 3 cell types are not, what I'm asking in an exam, for example, then they have to come from the other lineage, right? So lymphoid lineage, T cell, B cell, natural killer cell. Everything else is myeloid lineage. Everything else what? All the other formed elements of the blood. Okay? Now let's take a look here at the myeloid lineage. You can have myeloid that becomes red blood cell. Okay? You can have myeloid that becomes platelet. Okay? So here it goes into reticulocyte and erythrocyte. Here it goes into this megakaryocyte. Look at the difference in the shapes. Okay? And they all came from the same stem cell. So this guy here is called a megakaryocyte. Mega because it's huge. It is big. And after it becomes a megakaryocyte, it is going to lose pieces of the membrane with some of the cytoplasm, and then this is what is going to become the platelets. Okay, so megakaryocyte gives rise to platelets. All right, so besides those 2 myeloid lineages here, then look at these guys over here. Okay, myeloid lineage is going to give you eosinophils, basophils, myeloblast cells, neutrophils, and then you're going to have monocytes. Also, they're all coming from this myeloid lineage. These are all big white blood cells. Okay, actually, all of these guys are white blood cells, right? But these guys here are very, very important for innate immune response. Right? Innate immune response, most of those white cells are coming from the myeloid lineage. There is one exception for all of the white blood cells that are important for innate response, and it's the natural killer cell. So this guy is also important for innate, but this is the one that comes from lymphoid lineage. Okay. All right, so different lineages of your pluripotent stem cell. So here will be a first question that we would get, but of course we're not doing the clicker today. So be ready for these questions to be like the first slides for Wednesday. Okay, so then you can come to class and get your points like right away. So question here, hematopoietic stem cell will give rise to, let's see, what are the options? First one is erythrocytes. Do they give rise to erythrocytes? Yes, those are red blood cells. How about leukocytes? Yeah, white blood cells. What about platelets? Yes, so your answer is all of the above. All right. All right. So here are some terms or some association with homeostatic imbalances, right? That can happen because of dysregulation in pluripotent hematopoietic stem cells. So one example here is acute myeloid leukemia. So when you talk about leukemia, you're talking about a problem of leukocytes, which those are white blood cells, in the blood. So emia has to do something with the blood, right? So leukemia is white blood cell stuff in the blood. This is cancer, right? But then what kind of cancer? Then you look here. Myeloid. Myeloid has to do something with the lineage of development of some formed elements, right? So myeloid is something that does not have to do with those lymphoid cells. So when you look at the myeloid leukemia, that has to do with abnormal formation of white blood cells of the myeloid lineage, okay? So when you have this type of cancer, you're going to produce a lot of abnormal white blood cells of this lineage, and the overproduction of these blood cells in the bone marrow is going to mess up the production of the other formed elements of the blood. Okay, so this is the main mechanism why blood cancers kill. Okay. Now, when you see this type of leukemia in a patient, mainly is going to be developed in adults. Okay, so myeloid lineage leukemia, you're going to find that most commonly diagnosed in adults. Here's another example of another type of blood cancer. This is lymphoblastic leukemia. So you look at the leukemia part, yeah, white blood cells in the blood. Now these guys here are from the lymphoid path, okay? So this here is also cancer of white blood cells, but now you are going to look at those cells that are part of the lymphoid pathway, mainly T cells or B cells, okay? T cells or B cells. Now I'm going to say, don't you have also natural killer cells? But those usually are not involved in this type of leukemia, okay? So T or B cells. And the patient profile is usually children or elderly people, right? So you're going to find this type from the lymphoid lineage, mainly children or older people. All right. So now you should start thinking, how do you regulate the production of these blood elements in the first place, right? So you have to think here, well, you have to always be producing those cells, because most of the cells in the blood, they don't really last for a long time. So depending on the cell type, they're going to hang around for a few days. Some of them hang around for a few months. So in case of red blood cells, they hang around for about 120 days. How many months is that? About 4 months. Okay, so every red blood cell survives for about 4 months, and then it gets turned over, right? So you always have to be producing new red blood cells. Now, how is it that you're going to produce red blood cells? Again, what are the red blood cells important for? Oxygen, I heard oxygen. Okay, who said oxygen? Yes, yes, carrying oxygen, right? From the lungs to the tissues. It's going to carry carbon dioxide from the tissues back to the lungs, right? So do you want to have a good supply of red blood cells? Usually, yeah. Remember, red blood cells is carrying oxygen to all the tissues in your body, including your brain, right? If you don't have enough red blood cells, your brain is not going to work very well, okay? So you want to keep a certain level of, a good level of red blood cells running around, right? So there is a hormone that stimulates the production of red blood cells, and the name of that hormone is erythropoietin, right there, EPO, okay? So we usually have a certain level of this hormone in the body. Now, males, they have a bit higher percentage of this hormone than females. So you can assume, obviously, they're gonna have a little bit more red blood cells than females do. Okay, you're gonna see why. Why? Okay. So here, look, erythropoietin increases the production of your red blood cells. Now, the erythropoietin is a protein and is produced somewhere. It's a hormone produced in a place, travels in the blood, and it reaches the target organ, right? The organ that produces erythropoietin, the organ is the kidney. So your kidneys are going to produce erythropoietin, right? If your kidneys do not work well, your level of erythropoietin is going to go down, right? That means if you have kidney failure, you're also going to have anemia, which is low level of red blood cell. Okay? So kidneys are going to produce erythropoietin. Now look at this, it's interesting. Testosterone, which is another hormone, testosterone stimulates the kidneys to produce more EPO, right? Now that should make sense because I just mentioned that men have more red blood cells than females, right, than women. Why? Where is it that you produce testosterone? All right, can you think about organs that produce testosterone? Testes, exactly. You should be thinking about differences between men and women, right? Testes. Yes, you also have ovaries and female reproductive system that produce some testosterone, but testes is right in the name, right? So if you have testes, you're going to have higher level of testosterone. That means you're going to stimulate your kidneys to produce more EPO, right? If you have more EPO, you're going to have more red blood cells getting produced. That means you're going to have a higher capacity to carry out, to transport oxygen and to the tissues and waste products out of the tissues, right? That means if you have more red blood cells, you're going to have a higher level of endurance, for example, when you practice some kinds of sports, right? Does anybody follow with the Olympic Games? Yeah, anybody watch the Olympics? Yeah, yeah. You see, in the Olympics, they have two categories of athletes. They have the male and the female, right? Why? Hmm? Yeah, it's not because men have more testosterone, but overall they can run faster, they have more endurance, they have more, they produce more muscle mass, right? They are just built differently than females, right? So if you have a certain level of red blood cells, just because of different hormonal levels, that means you can perform better than the females, right? And then, based on this blood fact, some athletes, back in the day, they figure out that if they get more blood, they can perform better, which is true, right? It makes sense, right? So certain athletes, up to a few years ago, they used to, look at this. This was very sneaky, okay? They go to their, exercise doctor, right? And they start to bank their own blood. So they're not donating blood. They take blood out, right? They keep the blood, right, in the fridge or frozen until it's getting close to competition. So let's say this is a high elite athlete and is going for Olympic Games. So just before the Olympic Games, he goes back to the doctor and he puts some blood back, right? So that is called blood doping. Anybody heard of blood doping? Yeah, okay. Now, this was, what they thought was clever because it's not a drug. It's your own blood, right? They could do something else. They could just inject themselves with erythropoietin. Just get the hormone and you produce more blood cells. But then, if you do a blood test, officials from the Olympics are going to detect that you are doing blood doping with erythropoietin, and that will disqualify you. Right? But doing, yeah. That I cannot give you an answer right away because I did not look into it. So the question is it true that menstruating women produce more testosterone? I would assume yes, but yes, logically speaking, yes, I would assume yes, but I will have to look into it to know a little better. But that's a good point too. Also, testosterone has to do with levels of libido, right? And then it gets interesting in the class because what does that mean? That means drive for some things that are interesting, like sex, for example, sexual activity, intercourse, etc. Women who passed menopause don't have a lot of interest, so most likely they have less testosterone. Yeah. Okay, so coming back to blood doping, right? So what happens now? Those athletes that did the blood doping, they're going to perform better. They have better endurance, better resistance, better performance overall. So nowadays, the committee for the Olympic Games, they will test, they will check out if athlete had blood doping or erythropoietin injections, or doping with erythropoietin, because that is an unfair type of strategy to win games, right? So erythropoietin, very, very important. Which one is the organ again that produces erythropoietin? The kidneys. What is the organ that produces testosterone? The testes. All right. All right, so here. Other things are also important for controlling production of blood form elements. Okay. Here is another hormone that is called thrombopoietin. Every time you see thrombo somewhere, you think about coagulation and you think about platelets. Okay? So thrombopoietin is a hormone that is produced by the liver, and that is important for stimulating production of platelets. By the way, the liver also produces important elements for coagulation. Okay? The proteins involved in coagulation cascade are produced by the liver. Okay. Other factors that influence production of form elements are cytokines. Okay, so those are small proteins that are produced from many types of cells. All right. Okay, so looking overall at the composition of the blood, so you have the liquidy part, that is your plasma, and then you have those form elements. And this picture is from a video from Jove. Okay, and here just continue on that Jove video. Okay. You can watch it. All right. Okay, so here you have your white blood cells, and here, look, only the white blood cells are complete cells, all right? Because red blood cells do not have nuclei, and then your platelets are just fragments, right? So white blood cells are the true cells, and they do have nuclei. All right, so here are more differences between males and females. You see that when you put the blood in a tube and you spin down the tube by centrifugation, you're going to have separation of blood components. So the part that goes in the bottom, this is what has your red blood cells. Then the part that goes on top, that's the plasma. And then you have this white little part in the middle that is called the buffy coat. Now compare the blood from a male to the blood from a female, and you see the female has more plasma and less percentage of that red blood cell separation. Okay? I'm going to see more into that. All right, so put the blood in the tube, spin it down, and you're going to have a bottom part that is made up of your red blood cells. Now, when you look at the proportion of your red blood cells to the proportion of the whole blood, you can calculate the hematocrit number. Okay, so look at this. This is a percentage of red blood cells per unit of the blood. Okay, so here you see that if you have this on a scale, okay, so you have this like number from 1 to 10, okay? So you see here the percentage of the red blood cell would be at 4.5, right? So this would be 45%, okay? Now, if you increase the percentage of your red blood cells to, let's say, around here, and now your percentage reaches about 6 or 60%, that means you're going to have more viscous blood, right? Because the red blood cells, they give more viscosity to the blood, right? If you look at the white blood cells, you see that they are not a big percentage of your total blood. Look at this. The white blood cells, they all get squished into this little buffy coat right there. this white little part right there, all your white blood cells get there. You don't really have a lot of differences. You don't have differences really, male and female on white blood cells. They're about the same, okay? But what you see the difference is in proportion of your red blood cells. So if you increase this percentage of red blood cells, you're going to increase your hematocrit count, okay? So if you go up to 60%, it's still okay, but you will start getting into dangerous levels, because increasing the red blood cells, you're going to increase the viscosity. If you make the blood more viscous, it's going to have a harder time going through very, very tiny capillaries that are close to your tissues. That means if the blood is too thick, it's going to get stuck, and you're going to start having problems of coagulation, okay? And that can be life-threatening, okay? If you lower your hematocrit count down too much, now you have opposite effect. You have very liquidy blood, and you're running through problems of anemia, not enough red blood cells. So that means you don't have enough cells to carry out oxygen to the tissues, right? And as we're going to see, a main cause of anemia is lack of iron in the diet, okay? And we also have issues with menstruating women, that when you have, there's like a lot of menstruation going on, you lose a lot of blood, you can become anemic, right? And then you have also complications of not eating a proper diet and menstruating. Then you are losing blood, you're not making up enough blood, you're low in iron, you get really anemic, right? Do you know what are the signs of anemia? How do you know somebody is anemic? Yeah. You feel cold, right? You look at the person is pale because you don't really have a lot of blood going around, so your body is trying to keep the blood to where is important, like your organs and your brain. You look under the eyelids and you're going to find this whitish, right? So that's a sign. Another sign is that the person is forgetting a lot of things. They can't really think right. And it's like, what? Is it like early Alzheimer's? No, it's just bad diet, right? Most of the time it's bad diet. Right? But okay, so I digress here. So the hematocrit number gives you an idea. Yes? Okay. That is an interesting question. There is this machine called the gravitron, something like that, that spins humans. So do you have that issue? Now you have to look at this whole system, okay? You are not going to have a difference in the density of the blood, but yeah, blood is going to go to the part that is, causing the centrifuge issue. And you can do simple experiments at home. Like, have you seen those magic tricks that the magician has a ball in one hand, and then somebody has their eyes closed, and he is like lifting the hand for some time, and then put the hand down, and then ask the person where is the ball. Basically, you can see from the veins, the hand that has less protruding veins is the hand that was up in the air. So it's not like the person was psychic, that they can just figure it out. But yeah, we can really go into a lot of discussion about how blood shifts around. We're going to get more into that when we go into losing the blood. Okay. All right, I do have like 4 minutes to go here, so I'm going to just start talking about the erythrocytes. All right, those are your red blood cells, and you're going to see that they have basically a donut shape, donut without the hole in the middle, right? So they are biconcave, and one important protein to keep the cell in this shape is this spectrin protein. It helps the red blood cell to keep that biconcave shape, right? So we pick up from this slide on Wednesday, Okay, so if you have any other questions, you can come up and ask.